Okay friends, in continuation of earlier discussion, in earlier video we have said that there is generation of action potential across the membrane of cardiac muscle cells, and this action potential, electrical potential, changes from -90 up to +20 approximately because of exchange of ions across the cell membrane. Dependent upon the opening and closing of ion channels which are present on cardiac muscle cell, we can plot the action potential curve for the cardiac muscle or cardiac muscle cell. What this diagram shows us, it shows that there are 5 distinct phases in the action potential curve of cardiac muscle cell. For convenience of explanation, they have been given names as 0 phase, 1 phase, 2 phase, which is also called as plateau phase, then 3rd phase and 4th phase. If we want to explain what is meant by all these things, then we can say that in resting stage, the cardiac muscle cell is having electrical potential of approximately -90 millivolts. This electrical potential gets depolarized as it comes towards positive charge when there is opening of rapid sodium ion channels present in the myocardial cell wall or cell membrane. Now, because of entry of sodium inside the cells, the electrical potential of cell is shifted towards positive charge. The cell becomes positively charged. Then, when it reaches to about +20 millivolts, there is activation of potassium and chloride outward channels, which are called as transient outward channels. Opening of these transient outward channels pushes out potassium ions from the cells, and because of that, for a small period of time, the heart muscle gets hyperpolarized again. Earlier there was depolarization, shifting towards positive charge. Because of outward potassium current, there is hyperpolarization or repolarization of heart. When this outward transient potassium channel currents get activated, this is followed by another current called as calcium inward current. These are L-type calcium channels which get opened and they remain open for a prolonged time. Now, whatever outward potassium currents were opened, they remain open, and there is balance between outward potassium current, which was responsible for hyperpolarization, and inward calcium current, which is keeping the heart muscle depolarized again. And this combined opening of inward calcium channels, outward potassium channels, create a phase called as plateau phase, second phase in the cardiac muscle cell action potential curve. After this, the inward calcium ion channel currents get blocked and entry of calcium, which keeps the cell depolarized, gets blocked, and then outward flow of potassium continues. This outward flow of potassium continues and cell gets completely hyperpolarized again. It comes to the range of -90 millivolts again. At this stage, there is continuous inward potassium ion channel current which is continuing. This is called as inward rectifying potassium ion channel. So this is how each cardiac muscle cell or the whole heart passes through these 5 distinct phases of hyperpolarization, depolarization, plateau phase, again hyperpolarization, and so on. So this whole curve explains what happens to the action potential across the cell membrane, cardiac cell membrane, in 200 milliseconds. This is the period in which the heart muscle once contracts. Now question comes, why there is depolarization at phase 0 again? sorry, phase 4 again. There is spontaneous depolarization of heart at phase 4, which happens in SA node, and for that there is a current responsible, which is called as funny current. Then before going to this funny current, let's see how ions are getting exchanged across extracellular fluid and intracellular fluid when there is change in action potential of the cardiac muscles. Initially in phase 0, there is rapid entry of sodium ions from extracellular fluid to intracellular fluid, and this is responsible for phase 0, a very steep rise in the action potential of heart muscle. In phase 1, there is activation of transient outward potassium and chloride ion channels, because of which the cardiac muscle cell gets hyperpolarized to some extent. At this stage, there is opening of inward calcium ion channels, which remain open for longer duration, and they counteract the hyperpolarization which is occurring because of exit of potassium ions from the cells. Okay, so the second phase is combination of these two events: exit of potassium from intracellular fluid to extracellular fluid, from inside the cell to outside the cell, and entry of calcium from outside the cell to inside the cell, from the surrounding fluid or sarcolemma. Sarcolemma word means the coating or sheath which is present on the cardiac muscle system. Sarcolemma, calcium starts entering into intracellular fluid, means inside the cardiac muscle cell. Now, after a specific time, this inward calcium ion channel current stops, gets blocked, and only outward potassium ion channel remains active. What is happening in this case? From intracellular fluid, potassium is being passed into extracellular fluid. So compared to extracellular fluid, now intracellular fluid is becoming negatively charged because it is losing positive charge. Okay? And this continues through phase 4. Now, in this exchange of sodium ion, potassium ion, calcium ions, there is role of different pumps which are involved, and these pumps include sodium calcium exchanger pump, calcium ATPase pump, and sodium potassium ATPase pump. Now these are actual enzymes and ion channels which are responsible for exchange of ions across the cardiac muscle cell membrane, and this is how action potential keeps on changing with each beat or with each passage of impulse from sinoauricular node up to ventricle. Just now I mentioned about a current called as funny current, which is present in sinoauricular node. Now this funny current is responsible for depolarization or generation of spontaneous impulse in the heart. Why it is called as funny current? Because this actual current is spontaneous leakage of sodium ions into the cells. This is slow influx of sodium ions into the cells, and this is actually a leakage. It is not any activation or inhibition of an ion channel or enzyme. And because of this leakage nature of this particular current, the scientists who discovered this particular current, spontaneous leaking of sodium ions into the cell membrane towards the intracellular fluid, they call this leakage current as funny current. And because of this funny current, there is possibility or there is ability in the sinoauricular node to generate its own impulses. This is how continuously there is change in the action potential across the muscle fiber membrane or cardiac cell membrane, which is responsible for depolarization, hyperpolarization, contraction and relaxation of cardiac muscles, leading to automaticity, rhythmicity and refractory periods in cardiac muscle cells. In next video now, we will see what is meant by cardiotonics and how they exert their action. Thank you.